Hello and welcome to this Australian Biocommons webinar. My name is Melissa Burke. I'm the Australian Biocommons Training Manager and I'll be your host for this webinar. This webinar is part of a series that has been developed by the Computational Community for Structural Biology Computing in Australia. And in this series, we've been looking into how deep learning can be used to design custom protein binding proteins. This series has been designed to inspire and help you recognize potential applications of these methods by showcasing some of the ways that the community is putting them into action. Before we begin, we will take a moment to pause and acknowledge the traditional owners and the custodianship of the lands on which we meet today. Today, I'm joining you from the lands of the Turrbal and Yagara people, and we pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. Today, we are thrilled to welcome Dr. Josh Hardy to the webinar as our speaker. Josh is a Senior Research Officer at WeHi and an expert in structural biology, especially cryo-EM. He has been awarded an NHMRC Emerging Leadership Fellowship, and he uses cryo-EM to study the 3D structures of proteins to understand how they work in human health and how mutations cause disease. He has a PhD from Monash University, focusing on the structure of human viruses, and has been at WeHi since 2020, where he has worked on the structures of pseudokinases, cytokine receptors, and microtubule-associated proteins. Josh is passionate about cryo-EM education and training. He founded the Training Electron Microscopy Processing and Optics program at WeHi, chairs the Ian Holmes Imaging Centre User Committee at Bio21, and is a member of the ARC Training Centre for Cryo-EM of membrane proteins. Today, Josh is here to talk to us about ProteinDJ, a modular and open source framework for protein design workflows. Welcome to the webinar, Josh. I'll now hand it over to you to start your presentation.
I'd like to thank the organisers for the kind invitation to present here, and I'd also like to acknowledge the Wurundjeri people, the Kulin Nation, whose land I'm presenting from today. So I'm very excited to tell you about a program that we've been working on for over a year, and this is work that's been done at WeHi. I'm from the Lucé laboratory, and this also involves the Pappenfuss lab. We've called it ProteinDJ, and it's intended to be useful to people getting into protein design, as well as people that want to use this on their high-performance computing clusters. So today I'm going to talk about protein design and give a brief introduction and tell you what ProteinDJ can do. I want to give some tips on getting started in binder design, take you behind the scenes for a tour of how we made the program, which might be interesting to some of you, as well as how to install and run ProteinDJ. So the second part is a bit more technical, but I'll start with protein design. So you've probably heard about it. It's actually quite a general term, and it comes in many flavors. We've been designing and modifying proteins for decades. This includes things like protein fusions, where we combine sequences, expression tags, solubility tags, other domains, or mutations, deletions, that sort of thing. And as you know from your probably high school or uni days, many sequence changes affect the structure. There's a sequence-structure relationship, and so this has an impact on protein stability and folding. And therefore, designing new proteins, also called de novo proteins, is the ultimate test of our understanding of the sequence-structure relationship. And I love this quote by Richard Feynman: "What I cannot create, I do not understand." De novo proteins have been around since the 90s. People have been playing with sequences and using computer algorithms to design new proteins. It was mostly alpha helical until 2003, when the Baker Lab made the first one with beta sheets. And you can see over the years, there's been many different types of proteins that have been designed, and the diversity is increasing. But more recently, what's happened is there's been this rise in artificial intelligence, specifically deep learning, that has accelerated de novo protein design. And this has been particularly around the release of Alphafold 2, as well as RFdiffusion, and recognized by the Nobel Prize last year. This field is rapidly growing in its breadth and depth. So it's a really exciting time to be part of this program. So I wanted to tell you what you can do with protein design specifically for protein binders. So this is a subgenre of protein design where you create new proteins that target other proteins, also known as binders. And as you can see here from this really nice figure, there is a lot of different applications for protein binders. And so they can be used to solve problems in biology, chemistry, and medicine and have really broad appeal. There's some examples here of antitoxins and antivenoms to protect individuals, as well as proteins which can tune signaling by binding to receptors or block viruses in cell entry, but also more advanced applications where we're looking at technologies like CRISPR, antibodies, and anti-cancer therapies. So there's really a broad range, and I think a lot of people have been interested in getting into this field and starting to make their own proteins. I mentioned AI. There's a rapidly growing list of software now that can do this. So RFdiffusion, but many others. And they can be broadly classified into two types at the moment. So the first is generative methods. And so this is where you train a deep learning model on the PDB database to generate new folds. And if you're using RFdiffusion, it's starting from random noise and it's denoising a protein fold out of that over time. The second type are called hallucination methods, and this is where you take a sequence, a random sequence initially, and you optimize it by doing repeated iterations of structure prediction. So you sort of repeat this again and again till the sequence evolves and forms a binder that has high confidence. And this has been made most popular through Bindcraft, but there are other ones since then. And so today I'm going to be talking mostly about RFdiffusion because that's what we've put in ProteinDJ, but I wanted to tell you about some of the other stuff that's out there. Some of these software, especially RFdiffusion, doesn't generate a sequence. So it only gives you a fold for the binder or a backbone. So we need additional software to generate that sequence. The most popular one is ProteinMPNN, which this can design a sequence to match the fold. And there's multiple variants that you may see floating around, such as soluble MPNN, ligand MPNN, etc. And the idea here is that we can design a sequence and then test that sequence using structure prediction. So MPNN will design a sequence for a backbone to try and recapitulate that fold. And we give that sequence to a structure prediction program such as Alphafold 2 to test whether that sequence will give us the same structure. So we can compare the output to the input, and that gives us confidence in the sequence itself. There are two challenges with binder design that I think are really important to mention. One is that it has a high failure rate. So when this was being done before deep learning methods, it required expression of hundreds and thousands of proteins to get one that worked. Nowadays, we have better structure prediction methods, and this has improved the chance of success, but you still need dozens of designs to test in the wet lab. And even for the structure prediction step itself, the success rate can vary dramatically. So it can be between 0 and 5%, depending on your target and the settings. So you have to generate hundreds and hundreds of designs to get enough to make it through the structure prediction to get to the wet lab and actually get a successful binder. I like this figure from this Nature Communications paper that shows two types of failure. The first is when the binder itself doesn't fold in the same way that it was designed to, so it has a different arrangement. And the second is when the binder doesn't actually interact with the target in the way that you expect or interact at all. So there's sort of two main types of failure for this process. The second challenge that's really important for people getting into it is that there's a lot of software to get your head around, and it feels a bit like a technology tree in a video game where you have to progressively unlock software to get to your end goal, which is to design binders. Many of the software requires installation of specific environments, and so it's a bit of a process, and I think Matt described it well. It feels like you need a black belt in Python to get these programs running. And even if you get it installed, it may not run efficiently. So we had a case where it's meant to use a GPU, but it actually was using the CPU and was running about 10 times slower. So, this can be, this is a real barrier, I think, for people getting into it. So in light of these challenges, we came up with an idea for a new program or an approach, which we call ProteinDJ after Dylan and myself. And it's something that we wanted to make that was easier to install, that was efficient in using resources, something that could be flexible and configurable, yet would give you consistent results. We wanted it to be open source and transparent, so you know what the program's doing, and it's easy to understand. And also that all the metadata is being captured and reported in a way that you can digest and process. And finally, and this was one of the hardest parts, was to try and make it modular so that we could incorporate future tools, because we don't just want a tool for now, we want to build a framework for the future, and incorporate new software as they come in. So I'd like to tell you what ProteinDJ can do, and then I'll tell you about how we made it. So ProteinDJ runs in 4 overall stages. So the first stage takes your protein target and it designs a fold for the binder using RFdiffusion. So this is, again, starting with noise and denoising to create a new fold. Then this fold is taken to stage 2, where a sequence is generated, and this can be 10 or 20 times, as many as you like, generating multiple variants, and we have 2 programs that you can use here. These sequences get passed to structure prediction stage, which is the 3rd stage. Again, there's 2 programs that you can use to do that structure prediction. And finally, structures that pass the structure prediction phase go to analysis and reporting, where it gives you additional metrics and information about your designs. So I'll explain what each of these does, but this is the overall workflow. So the first step is very exciting. It's when RFdiffusion will create new binders. This typically takes 1 to 3 minutes per design, so sort of scales with the size of the binder and the target exponentially. And you can see here on the right from Dylan's thesis is a huge variety, and it's really cool to see what comes out of the program. But what we found when we were using it was that it's a great program. It's very flexible, but it's almost overwhelming in its complexity. So we just decided to simplify it and streamline into 4 different ways of using RFdiffusion. So these are kind of different types of designs. So we've got de novo mode, fold conditioning mode, partial diffusion, and motif scaffolding. And I know those terms may not be familiar to all of you, but I'll explain them now, and hopefully in a way that makes sense for maybe some of your projects. So the first is de novo mode, and this is what RFdiffusion is famous for, right? It's for creating things out of nothing, starting with noise and diffusing a binder, and this can be near specific residues, also known as hotspots, or it can be in sort of any location. So here you've got an example of a protein with hotspots, and you can see here gradually it denoises and diffuses a binder. So you can use hotspots. You can also try without, and I would recommend doing both, because it is interesting to see where RFdiffusion wants to go as opposed to where you think the binder should go. So it's always fun to sort of put a new structure in and see what happens. But that's the sort of the most basic mode of using RFdiffusion. The second mode is fold conditioning. And so unlike de novo mode, which is we're sort of creating from this deep learning model, random folds, this is using specific fold and secondary structure information. So this is a popular approach before RFdiffusion, to start with a template, a fold that's kind of tried and tested, and trying new sequences there. The advantage of doing it here is that you can get fold diversity and get different types of folds. So here I have 4 example folds, and these are sort of like gold standard mini binders that have been very successful in the past. And so I've collated these folds from the Baker Lab to sort of plug into RFdiffusion, so you can get that variety. And I want to show an example of how this can be useful. So this is one of our benchmarking targets. It's HA, a protein from influenza A, and it has a very low hit rate. So when we do our tests, we typically see around 3 to 2% success rate for structure prediction. And you can see that when we use the de novo mode, the only ones that succeed are helical. So we don't see any successful binders with beta strands or beta sheets in them at all. Even when we use the beta model that's specific to try and weight the diffusion towards that. But when I use the fold conditioning mode with one of the beta strand models of ferredoxin fold, I get successful binders with beta sheets. So this is a way of sort of baking in diversity into your binder set, which I think is really kind of useful for going to the wet lab with different types of mini binders, because I'm hesitant to sort of go all in on one sort of fold for a particular target. The third and fourth mode are for when you already have binders or part of a binder. So If you've got a binder that you want to optimize, and this is very common when you have a de novo binder that is maybe suboptimal, the metrics are okay, but you want to improve the prediction confidence, you can take a design and put it through RFdiffusion and apply a bit of noise. So you're not completely noising and destroying the structure, you're only doing this partially. And so if you look at this diagram here, if we go all the way to the full sort of diffusion and full noise, we're basically going to get a different protein. But if we stop halfway and denoise that, we can get a variation on our input fold. So this is a way of generating slight variations of what you do, depending on how many time steps you choose and building diversity. And the fourth and final mode is motif scaffolding mode. And this is the most complex, but this is for when you have part of a binder. So you may have already a binding motif, it could be a peptide or a helix or something that binds your target protein. You want to make that builder and extend it bigger and extend that. Or you might have a scaffold and you want to put a motif into that to bind your target. So this is obviously requires very careful placement of the motif and scaffold parts, as well as specifying where you want the insertions and deletions. So that's why it's a bit complicated. But I think it has its place in protein design, especially when you may already have like a biological binder that you want to extend, for example. We noticed almost immediately that RFdiffusion sometimes produces some crap, and this is normal with diffusion type models. If you've ever used any AI art generators, you'll understand. And so we added a filtering stage, which allows you to filter out unwanted designs or low quality designs at an early stage in the pipeline. So things that maybe have one helix that have very large radius of gyration and not compact folds, you can get rid of them quite easily by using the filters that we've put here. So that's the end of stage one. We've got our backbones. We need to generate a sequence. And to do this, we're going to generate multiple sequences per fold. So we're going to really expand the number here of structures that we're working with. Typically, you would do 10 to 20 attempts per fold. And in ProteinDJ, we've put in two methods. So there's two programs that you can do to do the sequence generation step, and they have their different sort of algorithms and advantages for doing this. So the first is ProteinMPNN, which is a very famous and established sequence design program. What we've done here is we've incorporated that with something called fast relax, which was designed to work in cycles with ProteinMPNN. And what it does is it relaxes the structure of the backbone in between designing a new sequence. So you would run MPNN, generate a sequence, relax the structure, and then have another go at generating the sequence. So it does improve success rates, but it is slow because this relaxation step is something that's not GPU accelerated. So it adds a few minutes. So it's really a compromise between quality and quantity. You can skip this, obviously, but we have found that it does improve the success rate if you start with the same folds. So that's one option. The second option is a much newer program that came out more recently. This is called Full Atom MPNN, and this builds on MPNN to iteratively design the sequence and side chains. So ProteinMPNN only does the sequence, doesn't sort of look at the side chain placement and I guess optimizing that. Full Atom MPNN will put in side chains. It will look at how they're sort of fitting with the target. It'll prune back anything it's not confident with, and it will go again and it'll go again until it completes the sequence. So it's GPU accelerated, which is great. So it's fast, but it doesn't relax the backbone. So it's only modifying the side chains. So this is different to the fast relax method. So yeah, they're the two options. And I was curious to do a comparison because Full Atom MPNN, for example, hadn't been used for binder design. So I wanted to test and see. I also wanted to look at, there's some different models for ProteinMPNN, like the soluble model and the vanilla model, which is the original one. So the soluble one has been trained on soluble proteins only, for example, whereas the vanilla one includes all proteins in the database. And what we observed was that the fast relax was consistently the best. It always improved the success rate over 5 benchmarking targets, but that comes at a cost of time. And Full Atom MPNN was inconsistent. Sometimes it was better than ProteinMPNN, and sometimes it was worse. So it was really hard to sort of anticipate whether it would be better or worse than ProteinMPNN. But I guess for your target protein, you might find one or the other is better, but it seems to be target dependent in that way. Again, like with the structure fold filtering for RFdiffusion, you can filter based on sequence. So there's a score for each program, and they're calculated differently. But I would say here that the value of filtering here is unclear at this stage. So we had Dylan did some analysis in his thesis looking at some of the scores and how they correlate with success for ProteinMPNN. But it was quite hard to decide on a threshold and a cutoff for what's going to fail later on. So we're still sort of figuring this out, but I guess an important step for us was to start capturing this information so that we could actually look at that in retrospect. So yeah, that's an open question at the moment. And the third stage is structure prediction. And this is really an essential step. Like I've said, this is how we test our designs. It's the most effective way to test things in silico. It also takes a significant amount of time. So you're going to find that in your run, this is going to be definitely a bottleneck. And we've built in two different software options again as well. So there's sort of so again, keeping with that modular approach of incorporating multiple software packages. So the first is Alphafold 2 Initial Guess, which has been used for a few years now to basically test designs. And how it works is that the design, which contains a binder and target, is given to Alphafold 2, but with limited information. So Alphafold 2 gets a structure for the target, which is the initial guess, but it's only given the sequence for the binder, so it's not given any information about the structure. And then Alphafold 2 predicts the structure, and we can compare the output to the input and see if the binder has folded in the same way, is the binder in the same position, etc. So we're using that structure prediction program to test this binder sequence. So this is not the normal version of Alphafold 2. This is a kind of a faster implementation. So the multiple sequence alignment step is largely skipped, but it's still quite useful and has been used a lot for binder design. And there's some key metrics too. So the PA interaction score and the RMSD between the input and design are two really crucial ones that you can use to filter by. I was really keen to get a different structure prediction program in there. I wanted to get something of the next generation, the Alphafold 3 era of structure prediction software. But I decided to go for an open source program called Bolts. And this is to sort of try and keep the software as open source as possible. It approaches the success rates of Alphafold 3. It's very close, but I think the open source is a huge benefit for us. So again, we're making a compromise here on accuracy for speed because we want to get through hundreds and hundreds of designs. We also design some additional metrics to calculate and compare the structures. It's a work in progress. There's some limitations for targets with multiple chains and lots of chain breaks, for example. So still working on this, but I think it's a cool thing to see, like a newer generation program for the structure prediction. For filtering, this is quite complex because there are a lot of metrics for structure prediction, but not all of them are as equally important as others. So I highly recommend reading this preprint. It came out recently. People have looked at 4,000 binders and gone back and done all the prediction metrics for those and looked at what were, I guess, correlating with success in the lab, in the wet lab. So that's a really good kind of retrospective analysis. So we personally have been using for Alphafold 2 initial guess the RMSD of the binder and the PA interaction score and the PLDT, and this is kind of what was published with the RFdiffusion paper. But yeah, the field is this is still an ongoing sort of debate about what metrics to be used. So I encourage you to read, but we've included all the options, so you're not restricted from that perspective. So yeah, I hope that's helpful. So you've done your structure prediction, you've filtered out things according to the metrics. Hopefully you've still got some structures left over. If you do, they go to an analysis stage. And this is relatively quick compared to the others. But what we're doing here is just adding some more information that can be really helpful for just to, I guess, break ties between highly ranked structures, for example. So things that you might look at include things, buried surface area, the delta G, the interface, and maybe all the wet lab considerations, like you might need a specific PI or charge of the binder. and also checking whether there's an extinction coefficient, because if there's no aromatics, it will be invisible in your UV spectrometer. So that's just sort of a quality of life thing. But this all comes out at the end in a CSV file to look at in Excel with all the PDB files. So we've tried to make that as easy to handle as possible. This is how the metadata file looks. So you've got a spreadsheet. Each line is an individual design, and you can see they have a fold and a sequence ID. And these are all the metrics that are correlated with those. So it's pretty easy to sort and filter and curate at the end. Yeah, so that's the end of the workflow. I thought I'd mention there's some limitations. So it's not suited for antibody design or design of enzymes, small molecule binders. These require different tools that we haven't incorporated. So there's RF antibody, there's ligand MPNN. So this is not, I wouldn't recommend using it for that, but it's great for protein sort of binding interactions. Bindcraft is not part of our pipeline yet. It does give different binders to RFdiffusion, so I'd recommend trying both. We're not here to sell a particular design program. And I also wanted to say that our workflow is linear, so it's not having an iterative cycle where the best designs go back through and are automatically recycled. That was too complex for me to program in. But you still have the ability to do that using the partial diffusion or motif scaffolding approaches. So it's just more of a manual process. And obviously, we haven't been able to test this on every HPC. We don't know what your environment will be like, and there are bugs that I'm sure will come up. So please send us an email or log an issue on the GitHub, and we'll try to address those. And through the feedback and testing from the community, we hope we will make it easier and improve the documentations and tutorials that are already there. So that would be great. Okay, so how do you get started in binder design? There's been some great talks in this series already, so I thought I'd throw in my 2 cents. And I think a really key thing is understanding your target protein. So obviously all this software stuff is only part of it. You need to understand the structure of your target protein and how it works, what you're trying to do with the protein binder. You will notice that when using RFdiffusion, it will have an affinity for hydrophobic patches. So some target proteins will be really easy to generate binders against, others will be much harder. And so in that case, if you find yourself struggling, try different hotspots and sample around the protein, different input structures as well. Or you might have to change and think about, can I do a homolog or change my target protein in the worst cases. It's very sensitive to the input PDB model. So it's kind of a quality in, quality out process. So look very carefully at the structure you're using. Is it a good crystal structure? Is it a good cryo-EM structure? Are you better off using a structure prediction program? There are unnecessary parts that you should remove, or maybe things that you've cut off that you should include, for example. So this is a very important step. The other thing that you'll see people do, because RFdiffusion runs slow with big structures, is cut their structures down. So you can trim it down to the domain of interest, but you have to be careful when you do this. I've got an example here of the insulin receptor, and let's say this is our domain of interest. Here is a nice clean cut where we've got sort of one protein sequence, and there's sort of a nice area for the binder to go to. But if we were to cut this domain in half, we'd have chain breaks, we'd expose internal residues, and RFdiffusion is for sure going to go and target those residues and give you binders that don't make sense in the real context. So you have to take care when doing this cropping. We've actually made a new functionality to put this uncropped structure back in to the program. So you can give RFdiffusion the crop structure, the small one, but in structure prediction, which runs faster, you can give it the full context. And so this is a way of providing more information for the structure prediction steps. So it doesn't slow down the whole process, but you still can ensure that. I have here an example of, so this is a trimer, and we've cropped it down and removed one chain, and the binder has gone and been designed in the interface that would normally not be accessible. But by providing the uncropped structure, we can see that it's flagged as problematic and it's not going to work in the full context. So this is another feature that we included to kind of make it easier for researchers, and a problem that a lot of people had mentioned to me about this sort of RFdiffusion size limitation. Good binders come in different shapes and sizes. I think we've seen in the literature lots of different Binders have been successful, and it obviously varies for target proteins. But in general, you want to have something that has a hydrophobic core, so it's got buried side chains that's going to help the protein fold. You need it to have shape complementarity with the target protein, that it fits into the surface and also has a decent buried surface area contact, and that may or may not include hydrogen bonds. But all of that is going to improve the chance of it being a high affinity and sort of successful binder. The other thing that's somewhat surprising is that bigger is not always better for binders. So you can have successful mini binders as small as 60 residues. So you don't necessarily need to design something massive unless you have a particular application in mind. So my recommendation is to try a range of sizes for your binders and see what suits your target. But yeah, you can use smaller ones. I think as much as the metrics and the CSV file and all those stats are useful, you should look at your best designs in Income ERX before ordering anything. You want to check that it's not clashing, put different structures in and sort of look at the context and think about it, flexible regions and all of that. So even things like MD analysis, I think will be useful in determining what's good. Although it's not my area of expertise, I think it is worthwhile to have an inspection of the designs. And remember that even if you do all this correctly and you have a really highly scored prediction, it is only a prediction, and your binder may not work, may not express or bind in the lab. So there's going to be a subset of designs that don't succeed. It can be difficult, I think, when you're starting out to figure out what parameters are useful and important. So I was doing this, I learned that there was different MPNN checkpoints. And so I was doing tests of those and comparing those for the 5 benchmarking targets. So you can see here, this is sort of 15 individual runs. And then I wanted to try different folds. And so you can spend a lot of time trying to figure out what's good, I guess, is a general case, but also for your target protein. So we sort of were experiencing this, and I said to Dylan, it'd be great if you could make a program to do this for me, where I didn't have to sort of set up 15 jobs and manage the progress. That would be really convenient. And so he designed a program called Binesweeper. And what this does is it searches across parameters. So you can set up different parameter combinations, and it will launch ProteinDJ jobs for each of them and give you the success rates. So you can give it a different sets of hotspots. You can give it a value here, the noise scale, and test different noise scales. Whatever you like, whatever parameters in ProteinDJ, you can run this and generate variations. So it's really good if you want to sample at a small scale first before doing a large scale run and sort of test those ideas. So this is a bit more complicated than the main interface. It only works on the command line. But I hope I think it's a cool way of sort of thinking about this problem. All right, I wanted to go into a bit more of the technical details of how the program works and also how we made it. Excuse me. So when I was getting into this, I didn't know anything about RFdiffusion. And luckily for me, there were people in Melbourne who were pioneering this technology around me. So that included Reese, Maria, Gavin, Cynthia, Margin, and Richard. And I know many of them have actually spoken in this seminar series, so I encourage you to watch their stories. And it's exciting to see work being published from these projects. And at the time, they were using 3 programs: RFdiffusion, ProteinMPNN, and Alphafold 2. So that was my first job, was to try and figure out how to install those. How do I get them running? And that was challenging for me. It took me about 2 weeks. The other things I found challenging was that sort of some programs used GPUs, some didn't. There was different metadata formats, they're producing files, and I was trying to grapple with those. And also there was, even for the structures, there was this thing called a silent file. I had no idea what that was. It's kind of a compressed PDB file. So yeah, I found it a bit confusing at first when I was getting into it. I had an attempt at a workflow. Over a year ago, I started to think about how I could tie this together. The only programming language I knew was Bash, which is a command line language. And essentially, this is a big Bash script that's running commands sequentially, just working its way through a process. I called it Big Script as a joke. But it had some major issues. So it only worked for me, and it only worked on the Weehai HPC, so it was no good to Gavin Reese and Monash and Bio21. It was requesting a GPU node even for tasks that didn't require GPU, so that's not very nice and makes your HPC manager upset. And it was difficult to access the settings, so they're in this massive file, right? And you have to, if you change target proteins, you have to go in and find the right parameters and tune this. So in general, it's very hard to use and adapt. But I could see the potential because it was able to generate 1,000 designs in 24 hours. So I could get lots of designs going, and it was parallelizing. And so, yeah, I could see the sort of potential, but obviously I knew what I'd made was not very useful. As I'd hit the ceiling of my computer skills, we had to reach out to others, and that's when I met Dylan. Dylan started honours in the Pappenfuss lab with Tony, and he had a background in software engineering. And so we decided to start an interdisciplinary project, and they suggested 3 software solutions. So first was Aptainer to containerize the software and make it work across different systems. Nextflow to tie all the steps together and make it into a workflow, as well as Python scripts to sort of help with glue the programs together and provide metadata processing and different steps like that. And so this is work that was done with our lab as well, involved Julie, Junshi, Andrew, and my supervisors as well, kind of supporting. So we had this interdisciplinary team. I think it was challenging to communicate about different aspects, but has led to a really useful tool, I think. So I had to upskill massively because I didn't know much about programming. And so I initially started just by modifying and usually breaking what Dylan had done in the first version of ProteinDJ. I went to textbooks, I read a lot of documentation, and I also used generative AI. So I use this a lot to not only develop and test code, but also to just ask questions about how the software were working. And I don't think video games are very useful, but sometimes it does feel like programming. But yeah, this was a really steep learning curve for me, obviously, try and get my head around the 3 different programming languages we're using. I wanted to briefly touch on coding with generative AI because this is actually how I learned to do this coding, and I think this is kind of how most people are learning now, using AI as part of the coding process. It's really good at understanding your intent and what you want. It usually picks up on that straight away. It will give you clearly annotated code and functions, often better than a human will. And I like that it gives suggestions for improvements and troubleshooting. I think those things are really helpful. However, it does assume that what you've given it to start with is functional and effective. So it'll rarely scrap what you've done and start over, even if that's more efficient. If you don't tell it that you're unsure, it will just assume that you're sure that your code works properly. Something we learned the hard way was that you need to test everything yourself and check the outputs, because often the programs will have these messages that will say alignment completed successfully or statistics calculated. But if you don't actually check those yourself using a different software package or downloading the structure and analyzing it, you can be sort of misled, and maybe the scores are incorrect, or that alignment hasn't happened, for example. So yeah, you need to be really careful about that. Also, I found that not all chatbots are equal, so there's quite a lot out there now. It can be overwhelming in choice. Comparing some popular ones, ChatGPT and Claude, I found Claude to be more reliable and more successful when I was running code. There's also these models called reasoning and thinking, and they can be really good for the more challenging tasks where you're thinking about, sort of the overall architecture and design, and less of that sort of small problem kind of conversations. So I use specifically Perplexity Pro, which is like a premium version of Perplexity that has access to the different models, so I could switch between them. So I recommend that if you're interested. The other thing I found in this process was that it does require a conversation, because you need to work through the code like you're working with another programmer. And in that process, it's better to start with as much detail as possible. So if you take the time to describe the problem, it will usually work much better. So I would do things like attach log files, show an example of an input file, and maybe an example of the output file that I wanted. And so it has that information to draw on, and it's better to spend more time there than sort of, an hour going back and forth and drip feeding information in. So I think that's something that helped me. And obviously, as I learned and started to sort of, understand what a Python script looks like and what the functions do, it helped me spot issues myself and also provide more practical suggestions for improvement. Because if you don't understand how the language works, it's hard to sort of say, describe what you need in a way that's practical. All right, so we use three things in our program. We use containers, Nextflow, and Python. So the first, containers, has I guess I'm using the analogy of organ transplants, but this allows software to move between computers in a way that's easy and doesn't break. It also allows programs to coexist that require different libraries, and this is quite common where you try and install two programs on the same computer, and they'll need different versions of software, and it can be really tricky. But by building them inside these containers, they have like everything they need within that environment, and also means that you can move them between computers or put them in the cloud. And so they're really useful. It can be tricky to get them going, but once you have them set up, they're very adaptable. So that was a key ingredient for what we made. The second is the kind of the architecture of ProteinDJ is using something called Nextflow. And this is a pipeline language that's compatible with containers and HPC and has been used a lot in bioinformatics for processing sequence data. There's been a move to using this towards for protein structures as well. So I think this was a kind of, a different application than normal, but we use this to connect all the programs together. So essentially, data and files work in channels, and so tasks can be performed on each file as they sort of flow through the pipeline, as illustrated in this metro style diagram. So you can set this up how you like. And the third and final ingredient was Python. And this is kind of for the little bits and pieces that we couldn't do in Nextflow. And this is the sort of things where you want to maybe align structures together or extract a sequence or something like that. So there's these great tools such as PyRosetta and BioPython that you can use that have this sort of functionality built in. And we use that a lot. And so there is one caveat with the ProteinDJ software is since it uses PyRosetta, you can't use it for commercial projects unless you buy a license for that. This is the same, by the way, for Bindcraft and some of the other tools out there. So you should be aware of that. And I did see someone sent me this recently. Someone's working on a free version of Bindcraft that bypasses PyRosetta. So I think that's interesting and worth checking out. So yeah, that's sort of the main ingredients without going into too much detail. One thing I wanted to talk about, which was, I guess, a big design challenge, was we have all these programs. They spit out different formats and files, and we need to figure out a way to get them to talk to each other. And this is a problem that took us a while to figure out because we tried different setups. So here on the right is an example of like one of our earlier versions. You can see it's very messy. We've got files coming from everywhere. Eventually, we settled on sort of some rules for how the metadata works. So we stuck to individual PDB files. Each structure has its own PDB file, as well as its own individual metadata file, and that's using a JSON format, which is very easy for computers to read. And in each metadata, there is an ID tag for the fold as well as the sequence. So there's two IDs, and these are in the PDB file names and the metadata. So that helps track which is which and make sure that the metadata is sort of mapped to the right structure. And we also use tags for the metrics and parameters. So these prefixes tell the user which program they relate to. So this is the mode for RFdiffusion. This is the temperature for ProteinMPNN. And these things are really nice even for programmers because it can tell you, what you're working with exactly. So yeah, we've kind of come up with these rules, and the whole point was to give us something that was interoperable. And so we have a consistent start point and end point for the module. So again, with this sort of modular strategy. This is how it looks. This is my attempt at a metro style diagram, but you can see it is quite complex. There's a lot of moving parts. I've highlighted the 4 stages in these gray boxes. So we have fold design, sequence design, structure prediction, and analysis and reporting. And you can see that all of these steps are spitting out metadata, and this is being collected in the final stage. So as the program is running, the metadata is being compiled and collected. So I'm not going to go into detail in this diagram, but this is essentially how it works, and you can have a look at our preprint if you're interested in more detail on how that all comes together. So I thought I'd finish by explaining how to actually get this running if you're interested in trying it out. So I tried to simplify the installation process as much as possible, so I reduced it to 2 steps. The first is obviously to get the code, so you need to clone the GitHub, which takes a few seconds. And the second is that you need to download models and checkpoints for things like RFdiffusion and Alphafold 2, because we don't own those. Obviously, we didn't make those programs. But I made a script to help you download that, so that takes between 10 and 15 minutes, depending on your internet connection. And once that's done, you have everything you need to run the pipeline. So our containers are in the cloud, and when you run the stages, it will download the containers for you and stage them. So you can build them locally if you want and have them in a central location, but you don't have to. That's how Nextflow is designed to run with these cloud container systems. You might need to tweak the resource requests. So if you have different GPU types, we obviously built it in our environment. So if you're using a different model GPU to us, you might need to change that. But it's pretty straightforward, and it's all kind of outlined in the documentation on our GitHub on how to change that. But obviously, you'll need to adjust that for your cluster and specific environment. In terms of actually setting up a run, you need to provide design parameters and also input files. So I mentioned the target, structure file. You need to have that ready to go. If there's any hotspots you're interested in, and essentially there's 2 ways you can start the program. So you can do it by the command line, or you can use a web server using the Secure service. So there's sort of these 2 methods of interacting. The first is command line, and I spent a lot of time on here, obviously, when I was developing it. But we've tried to make this as clean as possible with sort of annotation and notes and everything like that. So Nextflow runs using a config file, and inside that has all the parameters. And so you can see here there's the 4 main parameters, and I've put notes above about what the different options are. And that will take that file and launch the run. You can also use these things called profiles, which are sort of snippets of parameters, and essentially will be override what's in the other part of the config. So it's a bit easier to manage because you're only focusing on the ones that you care about. And I've got examples there for each of the different design types that you can sort of start with and tweak. And that's how it looks while it's running. So Nextflow will track all the jobs and tick them off as they're complete. So you'll get a progress update, and at the end, a summary of the number of designs and what's actually happened. So it is an easy way to use that, but I understand the command line is not for everyone. So I also wanted to build in functionality to interact with a web server. So Nextflow, there is a premium service called Secura, and this allows you to run Nextflow pipelines in the cloud or on your HPC. So here we have ProteinDJ running through Secura. So you've got all of the different design options. There's a custom form for each one that will have default values, and it will check your input and make sure that your input's in the right format. So it's a lot easier to use, I think, than the command line. And the other nice thing is that you can see all the jobs that you're running as well, and it will give you that progress update, but you can see multiple jobs at the same time. So here at WeHi, we've partnered with Oz Biocommons, so we have access to this service. But if you're interested, I highly recommend getting in touch with them and seeing if you can set that up, because we've been finding it really useful. To wrap up, I think that over the course of this project, this program evolved. I think it's a really useful tool for now, but we've tried to make it as much as possible into a framework we can use for the future, because we know that the software landscape is rapidly evolving. There's a need for conventions, interoperability. And so all our code is open source to help that process and enable collaborative efforts to improve this further. I'd just like to briefly thank everyone who was involved in the project, especially Dylan, who is the brains behind ProteinDJ, and the initial development taught me a lot of coding and things like that. Very patiently put up with my naive questions, is a supporter of this project, and mentored me throughout, which was a big help. And Andrew and Junshi were really important in sort of, I guess, beta testing the software and giving us feedback on how all the parts came together, and the rest of the Lucent lab. Julius Kander did a massive job setting up all the HPC environments and fixing some of the containers to make it work. Really grateful to her, and also Tony as well for supporting the project and guiding us through that with WeHi Research Computing. I'd also like to acknowledge all the members of the protein design community that I've interacted with over the past year, and for all the conversations and advice. And if you're interested, you can check out the code in the bioarchive on our GitHub. And thank you very much for listening. I'll take any questions.
Thanks very much, Josh. It was a really fascinating talk. And I can see that ProteinDJ is really going to improve the accessibility of these tools while still maintaining the flexibility of them as well. So we do have time for questions now. If you have a question for Josh, please put that into the Q&A box. And I'll also just flag that we have pasted the link to the preprint as well as to the GitHub into the chat, so you can go and have a closer look at those. We'll also be sharing the slides after the webinar as well. So Josh, I think we'll ask with a question about, start with a question about the development of ProteinDJ. And the question is, how did you decide which programs should become part of this workflow?
Yeah, that was a difficult choice. Each program took us weeks to months to really, I think, program properly. We had to make sure that we understood how they were working, and sometimes we had to modify the source code as well to make sure that it was fitting in with the pipeline. So it was quite difficult. I think we went for, like I said, the open source software had a huge advantage for us, so we tried to stay with that as much as possible. But yeah, we had to, I guess, have a go at different software packages, and that process is ongoing as we sort of look at what's out there and think about what's next to incorporate into the pipeline.
Definitely a fast-moving field. If I've learned one thing from this series, it's that there's new things coming out all the time. Okay, turning now to a couple of questions from our audience, and these ones are more about the binders themselves. So first question is, in your experience, do the beta strand containing binders give you increased solubility and protein stability?
So I haven't actually made many binders myself, so I can't speak to sort of, I guess, the success in the wet lab. I think that you have, I guess, examples in nature of both. So I think one of the things I think about is if the sort of algorithms at the beginning are not producing folds that have these elements, then they don't make their way into the sort of testing in the lab. So I think there has been a bias towards helical folds in the past. There's a paper, I think it's from 2017, 2018, that was looking at, from the Baker Lab, looking at the different types of folds and they basically make synthetic proteins. And they observed that the small helical bundles were more soluble than the beta strand containing proteins, but you still have successful cases for both. So it's not mutually exclusive.
Thank you. So another question from the audience. This one's about reverse engineering the process, and they want to know, has anyone tried using the de novo design binder to redesign the target?
Oh yeah, that's interesting. Yeah, I'm not sure. I feel like every day on my email watch list, I get stuff from the Baker Lab where they've done something wonderful and exciting with protein structures. I think there's, I guess, one of the reasons that I wanted to try incorporating bolts into the pipeline was to co-fold the target and the binder together, which I think is sort of an interesting way to think about these interactions in proteins. And I'm also keen to try Bindcraft because that does the same thing at the earliest stages. So yeah, there is this kind of, I guess, induced fit that can occur. I think molecular dynamics will have a big role to play into sort of designing protein binders to induce a particular conformational structure of a target protein. I think that's something that will be very important. Unfortunately, I don't know very much about. Yeah.
Only time will tell then. Okay, one last question to wrap us up for today. What's next for ProteinDJ?
Yeah, so we are obviously still working on the program and making sure that it runs. So making sure it runs on different environments and tidying up the bugs, but also incorporating new software. So we want to incorporate things like Bindcraft and also improving the structure prediction metrics. So that paper I mentioned that came out recently has some new formulas for deriving some metrics that are really powerful. So we want to incorporate those into the pipeline. So building on what we have and adding that in. So yeah, that's probably what's most soon.
Great, thank you. Well, we are going to have to wrap it up there for today. If I could just take the screen share back for a few final housekeeping things. Thank you. As I mentioned at the beginning, this webinar is part of a series of events that we've been running on this topic. There are a couple more coming up. You can find out more about those and other events that we are running on our website. You can also find out about the Australian Biocommons Secura service there as well. If you want to know more about the Australian Structural Biology Computing Community, you can do that through the link that you see there on your screen, which is also going to be put in the chat in a minute. And this is a really good place to connect with other people who are doing this work through their Slack group and through the regular meetings. So if you are just getting started in this, I encourage you to go and explore that community. Finally, thank you again, Josh, for joining us today and sharing the story of ProteinDJ with us. And thank you to all of you for joining us as well. As we leave, I'd like to acknowledge that Australian Biocommons is enabled by NCRES via Bioplatforms Australia funding. Thanks again for joining us. We hope that you've enjoyed this webinar and we look forward to seeing you again soon. Until then, goodbye for now and enjoy the rest of your day.